All right. So next we have our second presenter of the evening, and that is Charu Singhal, and she is the co-founder and CEO of a company called Etta.
Hi, Katie. Thank you for inviting me here to pitch. Shall I share my screen?
You absolutely can.
Perfect. Hi, everyone. I'm Charu, the CEO of Etta. We're an AI company that specializes in building visual technology that can help detect tumors that are challenging to see with the naked eye. Thanks so much for inviting me back to pitch again. The last time I was here, I pitched our AI-Dermatology tool in the Dermatology Pitch Club. Today, I'm pitching a new product for a specialty that we've expanded to. White light cystoscopy is used to detect cancers in the bladder. One of the most challenging cancers to detect is carcinoma in situ, also known as CIS. This is a non-muscle invasive bladder cancer that is challenging to see for even specialists due to the fact that there may be little to no contrast between the tumor and the surrounding areas of the bladder. Non-muscle invasive bladder cancers are cancers found on the tissue that line the inner surface of the bladder. 75% of bladder cancers are non-muscle invasive, and half of the patients treated with transurethral resection of a bladder tumor have a recurrence of the disease. While CIS is challenging to identify, the identification of CIS in a timely manner can have a major impact on follow-up clinical decision-making, including surveillance, cystoscopy, post-BCG treatment, and XRT patient selection. Currently, in order to detect CIS, there is a method, tool, and process called blue light cystoscopy. The company that builds the tool for this is called Cysview. This is a tool that has a high startup cost and a formula that must be rendered into the patient's bladder that can take 45 minutes to absorb. Not many hospitals utilize blue light cystoscopy due to the low reimbursement channels, the high cost to start up, and patient hesitation. At this time, there is no routine screening in the general public for bladder cancer for individuals that are at high risk for bladder cancer. A tool like this could provide support to a specialist at a diagnostic level and make screening more likely to provide early, accurate information. We are building a solution that overlays our AI real-time identification technology into the white light cystoscopy process to augment the intelligence of urologists by helping them identify CIS tumors. Here's how it works. As you can see, we're in the OR, and urologist here has inserted a cystoscope into the patient and is examining the bladder for anomalies. Here, our Etta tool has detected that there is a tumor inside this patient's bladder, and it is matching closely with CIS, as seen in our database through the models it is trained with. The urologist will likely perform a biopsy at this point. Although most hospitals do not have blue light cystoscopy available, all that conduct bladder cancer examinations have access to white light cystoscopy. As a company that values healthcare access and equity, we built a tool that provides the same ultimate utility as blue light cystoscopy, but simply uses AI overlaid into the white light cystoscopy that clinics already have. This results in no change to the existing workflow, no time added to a patient visit, and no discomfort for the patient. We started our company 3 years ago and have been achieving traction through a dermatology platform that helps clinicians identify, score, track, and map rashes and lesions. We found that some of the work that we did in scoring atopic dermatitis allowed clinicians to distinguish between texture and low contrast differentiations and nuances of depth within a lesion. We decided to apply similar methodologies to CIS in the bladder, and we're finding meaningful results in our early stages of testing. We layered a depth model with an edge detection model and an object detection model. The depth model differentiates the layers and textures within an image by comparing frames with their respective distances from the camera and the scope. This assesses the spatial structure of the individual's bladder from the vantage point of the scope, which can help a tool identify subtle distinctions in the contrast of the frames. We then layer it with an edge detection model to assess the texture and textural differences that could be shapes and anomalies that are related to CIS tumors. Finally, we have our object detection model that is trained on biopsy-verified CIS frames and from within bladders and white light cystoscopy. All of these models layered together build a tool that is an AI performing in real time more methods than the naked eye is able to perform. With just 100 CIS images and around 500 frames of normal bladders, we are able to achieve 88.9% precision, 72.3% recall sensitivity, and an F score of 79.7%. In cancer diagnostics within data science, these are the metrics that are considered relevant when validating a model for diagnostic purposes due to the high risk of the nature of a model missing a cancerous tumor. Precision tells us how many of the cancer-tagged outcomes were actually containing CIS tumors, and recall, which is also referred to as sensitivity, tells us out of how many frames were given, how many of the frames that had cancerous aspects were accurately tagged as having those cancerous aspects. The F1 is considered the harmonic mean and an overall measure of how accurate a model performs. Given the extremely low amount of data that we started with, these results are very promising, and we are certain that with a bit more data and training and testing, we would achieve a very high level of identification that could truly change the landscape of CIS and how quickly it is managed. As we mentioned before, we have an existing AI dermatology platform that has achieved significant traction in the marketplace for the last 3 years. We last pitched our full body real-time skin cancer product that is currently in pilot study at a research hospital based in Colorado. We wanted to expand our imaging technology in a place where it could be comparingly meaningful, and thus CIS was found to be similarly challenging in terms of visual identification. To roll this out, we are in the process of partnering with health systems that can collect more footage from the operating room, and we will use that to load up a clinical trial version of our technology. We'll conduct a trial based on the ethical guidelines from the UKCE, and we'll partner with a Cystoscopy company to scale up via a hardware pathway. And then we would like to see EdAssist utilized in every cystoscopy procedure. None of this would be possible without our amazing team of experts. We've always been backed by some of the most brilliant dermatologists in the country who helped us start and build some of the principles around our visual technology. We're now joined by Dr. Nathan Yarum, a urologist at a research hospital, who is helping us validate and bring this vision to life. We are so excited to make CIS tumors easy to identify in clinic, and I look forward to your questions. Thank you.
Great. Sorry about that. I was trying to put myself back on stage, but I think Mike got me taken care of, which is fantastic. Thank you so much for coming back and presenting on Etta again. I'm really impressed, by the way, since the last time you were here, with the traction that you have really been able to garner with the Etta Dermatology product. Tell us more about that, and then what made you think that we could take this technology and apply it to bladder cancer?
Yeah, absolutely. So, we've been around with Etta for about 3 years. We had a whole, we're as a founding team, we're engineers. So we were product people who teamed up with dermatologists. And we got to have like a really kind of fun pathway to finding product market fit, where we started off in a consumer space, helping people identify their own skin cancers, and then we took a telehealth angle where we were like the mediary between, seeing something on your skin and then connecting with a dermatologist, and then we took it to the ER with rashes, and then we took a pharma angle. So it really has been, dermatology was so much fun, to start up. And then in that process of evolving our product, learning the channels for how to validate a product, the correct way in the medical technology space, and how we were able to do that with dermatology, we felt like we were in like a good, safe space to kind of expand into other specialties and really do what we do best, which is visual identification.
So to clarify, and I'm just pointing this out because there's a couple questions in the chat about fitting into the clinical workflow. But with Etta Dermatology, that was not simply, but software that could be used on a tablet, right? And in the office, in the emergency department, or at home to be able to do a skin scan, etc. When we're talking about the bladder cancer evaluation, go talk a little bit more about the clinical workflow and how you talked about partnering with hardware producers. Talk a little bit more about how you're going to integrate that into the clinical workflow.
Yeah. So as a company, we're really excited about sort of innovation being, less is more, where you use less expensive products, but you build software into existing tools. I think, in this kind of a world that we live in, that is how we can achieve something close to health equity, is by, doing scalable things like building technology that can really provide support. So, when we talk about partnering with healthcare or hardware companies, that would be like Cystoscopy companies, to embed our software into their existing platform. And when it comes to the workflow, we know how hard adoption is in clinic, and putting in, another piece of hardware, especially if it requires a drug injection, education, patient, if you have to convince a patient to do it, there's so many barriers to entry that we think just, embedding software into an existing tool, and partnering with that tool is just, a much faster, more clear-cut way to scale.
Well, and I can tell you on behalf of many of the physicians that are here tonight, putting a new technology into the existing clinical workflow is absolutely the way to go, and I know you know that too. A couple more questions from our audience. So with a precision at 88% right now, biopsies clearly will still be needed, which you mentioned in your talk. How do you propose to improve the identification? Could you get to a point where you don't need biopsies or other major interventions for your patients?
Yeah, absolutely. I think as it stands right now, you eventually do need to do a biopsy. What we could do is limit the number of unnecessary biopsies. I think that is a realistic target for us. Changing the workflow on the end where they're not conducting a biopsy, that might be a more difficult sort of pathway for us. But reducing unnecessary biopsies is a big one. Knowing where the cancer is our current goal, is to help them see the CIS tumors that you are very difficult to see. But after that, it would be reducing unnecessary biopsies. And then, maybe long term there will be a better, but for now, I would say the standard is to always do a biopsy eventually.
Yeah, and I think my understanding is you're looking to partner with some urologists or uro-oncologists, really, because the more you feed your algorithms, the better that precision is going to get, right? So more and more studies where there is CIS involved, the better it's going to get, because of course it's AI. Is that right?
Yeah, absolutely. Since we are just starting out with this expansion, we do need more data. So, any urologists that are interested in being data partners, any hospitals that are interested in doing that, the more we have, the faster we can, take something like this into market.
Awesome. And a couple more questions from our audience. One, are you able to accurately detect margins and be able to help guide definitive treatment, if that's the case?
We're not focusing on that just yet. That is kind of a next steps. We're starting with just CIS, and then we're going to validate that and then continue from there.
Awesome. And I just have one more question, because I know you talked in the beginning about, and Mike actually put this in the Q&A as well, about the blue light cystoscopy. So we know that there is an alternative, a viable alternative out there right now. How is what you're doing an improvement over that?
Yeah, it's the cost to have it is around 116,000 for the hospital, and then there's like a 5,000 cost ahead of that. It's not reimbursable by most insurance, including Medicare. So that is like 3,000 out of pocket for the patient. It's very expensive, and I think it was like a great pioneer for CIS tumor detection, and kind of led the way into showing us that, like, hey, this is needed. I think that also, like the drug injection, there's so many barriers to hospitals and health systems with blue light cystoscopy, but it is great. We just see it as kind of like the first step, and then we're like the next stepping stone where it's like you don't need to do all of that. You can just use software overlaid into your existing hardware.
Wonderful. Well, Charu, thank you so much for being here tonight. I know there's still a couple of questions in Q&A, but guess what? Charu and Mari Lore are going to join us for networking after we finish up here. And you, I hope all of you and our audience will as well, because they are ready to take more of your questions, which is really exciting.